Synaptic transmission, or communication between neurons, has a number of different steps. We're going to describe the structure of synapses, how neurotransmitter is released, activation of receptors, postsynaptic potentials, neural integration, termination of postsynaptic potentials, autoreceptors, and other types of synapses. Let's start with the presynaptic neuron. This is the neuron that's going to be releasing neurotransmitter. It's the one doing the talking. There are a number of stages of synaptic transmission that happen behind the scenes. That is, apart from the communication within a neuron, separate from the action potential, or AP. Behind the scenes, neurotransmitter has to be produced and packaged into vesicles. And many vesicles can dock at the membrane release zone. This is what docking looks like. We see all of these different proteins tethering the vesicle to the terminal membrane. Neurotransmitter production is a little bit different for all of the neurotransmitters, and we'll talk more about that when we cover the specific neurotransmitters. The process of packaging neurotransmitters into vesicles is done by a pump, a vesicular neurotransmitter pump, that uses ATP to pump neurotransmitter into the vesicle so that we get a high concentration of neurotransmitter that can be released when the vesicles fuse. Vesicle fusion happens in response to the action potential. The depolarization of the action potential, or AP, when it reaches the terminal, triggers voltage-gated calcium channels to open. Calcium then enters the terminal. It's important to think about which driving forces would push calcium into the presynaptic neuron. Once calcium is inside the presynaptic terminal, it acts as a signaling molecule. It interacts with all of these proteins, pulling the membrane of the vesicle into the membrane of the terminal, causing this omega shape and ultimately allowing neurotransmitter to diffuse into the synaptic cleft. The postsynaptic neuron is the one listening to what's being said by the presynaptic cell. There are a number of things that happen behind the scenes. For example, the postsynaptic neuron has a lot of proteins at the membrane that make it available to receive neurotransmitter. That is, there are ionotropic and metabotropic receptors present at the synapse. And inside the postsynaptic membrane, there are second messengers, effector proteins, and other ion channels available to respond to the neurotransmitter. Once the neurotransmitter enters the synaptic cleft, it's going to bounce around and interact with whatever it bumps into. If it bumps into an ionotropic receptor on the postsynaptic cell, the neurotransmitter binding causes the ion channel to open and triggers a postsynaptic potential. If it binds to a metabotropic receptor, it causes a conformational change and triggers an intracellular signal transduction cascade, or STC. In this figure, we're looking at an axosomatic synapse, that is the axon of the presynaptic cell making synaptic contact with the soma of a postsynaptic cell. This postsynaptic density got its name because when you look at a synapse through an electron microscope, we see a dense area on the postsynaptic membrane. This density is caused by the high number of proteins that are present here. The terminal button is shown releasing neurotransmitter, and we also see other vesicles making their way down towards the release zone. Here's a cross-section of a synapse from an electron microscope, and again we can see synaptic vesicles and this omega figure, which is a vesicle that has fused with the membrane. It's important to know what a synapse looks like in general. In this lovely drawing that I did, we can see a number of different parts of the presynaptic terminal. For example, we have a vesicular transporter, which uses ATP to pump neurotransmitter into the vesicle. We also have a reuptake transporter. Not all neurotransmitters have reuptake transporters, but many do. These transporters also require ATP. We can also have presynaptic autoreceptors. These are activated by the neurotransmitter released from its own cell and serve as a negative feedback mechanism. On the postsynaptic cell, and in this case I've drawn a dendritic spine, so this is an axodendritic synapse. On the postsynaptic cell, we have a number of metabotropic and ionotropic receptors. Another important piece of the presynaptic cell is the voltage-gated calcium channel. This is what opens in response to the action potential reaching the terminal, allowing calcium to enter the cell and facilitate vesicle fusion. Upon entering the synaptic cleft, one of the things that neurotransmitters can do, and indeed the purpose of having released neurotransmitter, is to bind to postsynaptic receptors. Again, these receptors come in two flavors, ionotropic and metabotropic. Ionotropic receptors are ion channels. They are triggered to open by the neurotransmitter. The neurotransmitter is the ligand. So these are a subtype of ligand-gated ion channel, also called neurotransmitter-dependent ion channels. Metabotropic receptors are not ion channels. They are 7 transmembrane domain receptors and G protein coupled receptors, not ion channels. These initiate a signal transduction cascade. This figure is showing the difference between an ionotropic and a metabotropic receptor. When neurotransmitter binds an ionotropic receptor, the channel opens, allowing ions to flow through. Remember, the direction the ions flow depends on their driving forces. In the case of a metabotropic receptor, this is not an ion channel. But when the neurotransmitter binds, it can cause a change in shape of the receptor that triggers changes on the cytosolic side, initiating a signal transduction cascade. One of the things that could happen in a very short signal transduction cascade is that it could activate a nearby ion channel. This is not an ionotropic receptor. It's not being bound by the neurotransmitter, but it is another type of ligand-gated ion channel. A second messenger produced as part of the signal transduction cascade can open this channel by acting as a ligand binding to the cytosolic side of the channel. Once that channel opens, the direction ions flow depends on their driving forces. This figure is showing what a metabotropic receptor looks like at a little bit of a closer level. The reason it's called a 7 transmembrane domain receptor is that there are 7 parts of the protein that cross the membrane. 7 transmembrane domains. On the part of the receptor that's in the extracellular space, we find the neurotransmitter binding site. On the intracellular side, in the cytosol, we see a G protein binding domain. This G protein binding domain is hidden up until the neurotransmitter binds. In response to neurotransmitter binding, the receptor will change shape and the G protein binding domain will be exposed. This initiates the signal transduction cascade. A signal transduction cascade is a series of steps between messengers and effectors. Messengers are molecules that trigger an effector to do something. An effector is a thing that actually does something. It could be an enzyme, an ion channel, a transcription factor. Signal transduction cascades can be quite short or can be quite long.